All right. Good. Okay, great. Well, thanks, Jason. Thanks so much for this invitation. This has been a really great conference so far, and feel really privileged to be a part of it. So we're going to jump we went from RNA to DNA. Now we're going to jump into the epigenome, and I'm going to talk today a little bit about how we could potentially use the epigenome to illuminate social pathways of aging. And I just want to acknowledge funding from the NIA for this work. All right, I'm going to grab my little laser pointer here. Okay, so just an overview for today. I'm going to briefly talk about what epigenetics is, and in particular, one type of epigenetic modification, which is DNA methylation, what it is, what it does, and how it's quantified, particularly in population studies. Then I'll talk a little bit about biological aging and the epigenetic clocks, which is one way to measure biological aging that has become really popular over the past decade. And then I will give an example of one of my studies that's looking at associations between socioeconomic status and epigenetic aging. And then we'll wrap up and hopefully have some time to talk a little bit about what's next in social epigenomics. So what does the epigenome do? Turns out we have this major DNA packaging problem. So we have 5 feet of DNA that has to fit into a nucleus with a diameter of 6 micrometers. That's 1/1,000,000th of a meter is 1 micrometer. So that's a lot. It's been compared to fitting Mount Everest into a grain of rice. So it's a really complex problem that the cell has to solve. And so what the DNA actually does, or I'm sorry, what the epigenome does, is it maintains proper packaging of the genome across cell division cycles. So here you can see the epigenome is partially what's helping the DNA wrap around histones and then further into chromatin fibers. And then that gets packaged even further into chromosomes, which then can fit neatly inside the nucleus of the cell. And because of this, epigenetic modifications have a regulatory influence on transcription because they're actually controlling which parts of the DNA are open enough so that they can be read by transcriptional machinery, so that that they can come in and read the DNA. If it's really tightly packaged, the transcriptional machinery can't get to the DNA to read it and create the mRNA transcript. So as I mentioned, the most well-studied form of epigenetic modification is DNA methylation. And this is just where a methyl group is added to the DNA molecule at a cytosine base. And when it's located in a gene promoter, this usually represses gene transcription. Not always, but usually. In mammals, DNA methylation is almost exclusively found in CpG dinucleotides. These are known as CpG sites. And all that is a C nucleotide followed by a G nucleotide in the 5' to 3' direction. So here on the DNA strand, which we read from the 5' direction to the 3' direction, you can see it's just a cytosine phosphate guanine here on the DNA strand. And there's a number of important characteristics of DNA methylation that are worth mentioning, especially for population researchers. As I mentioned, it controls which genes are active, which is, the most important. But it also, with that plays a role in several key cellular processes, including cell differentiation. So DNA methylation is actually variable across tissues and cells. And for this reason, it's really important to think about, what tissue you're using to profile epigenetics in, and whether that tissue is a good proxy for the phenotype that you're studying. It also plays a huge role in aging, which we'll talk about today, and cancer. The epigenome is highly influenced by the environment, and I think this is why it's been really attractive. Way for social scientists to kind of bring in biological data to their studies. But it's also important to note that it is also under genomic control. So methylation is heritable, and there are sites on the genome that influence methylation. Another important thing to mention is it's modifiable. So, this is something that pharmaceutical drug companies are really excited about, and you can see why. I mean, if we can change methylation patterns in the genome to influence transcription, that could have huge downstream consequences for disease and aging and cancer research. Also, you know, as was kind of alluded to earlier in the talks today, it's not as sensitive to ancestral differences. So we don't have this same population stratification problem where we then have to really just look within ancestry groups. And so with epigenetics, people more commonly pool the data across ancestral groups. So how is DNA methylation measured and quantified? In large epidemiological cohorts, it's typically, you know, we use microarray data. Whole epigenome sequencing is just still too expensive. And with the methylation bead chip, that's the 450K or the EPIC, what they do is they use fluorescent probes to quantify the percent methylation at a given CpG site. And the EPIC array captures around 850,000 CpG sites. And the common data output here is just the percent of methylated cells at a CpG site. And this is known as the beta value. This is what researchers most commonly use and kind of, you know, what we'll be using today when we construct the clocks and talk about the clocks. So it's long been known in geroscience research that, you know, chronological age is a really imperfect measure of aging, right? Some people age faster than their chronological age, some people age slower. You know, chronological age is linear, but aging is not a linear process. And what's neat about the epigenome is that it actually exhibits really precise transformations with age. And because of this, about a decade ago, Steve Horvath and other scientists started to develop these epigenetic clocks. And what these are just composite scores of CpG sites that are highly associated with chronological age or other phenotypes of aging. And basically, they're using machine learning, supervised machine learning methods like ENet or Lasso to select these CpG sites that are most highly associated with whatever outcome they're predicting, in this case, usually chronological age or other phenotypes of aging. And as it turns out, these clocks really accurately predict chronological age. And if we look at the residual, so if we regress out chronological age, the residual, you know, which is known as epigenetic age acceleration, so how much, you know, someone is aging epigenetically above or below their chronological age is also highly associated with a number of age-related diseases and mortality. And, you know, I can't even begin to cite all the papers that, you know, have contributed to this science over the past decade. So I just have two review papers down here, which are really excellent if you want to dig deeper. Recently, Morgan Levine and colleagues published a paper that showed that, you know, actually these clocks, there's about 13 clocks so far, I should mention, that have been constructed in the literature. And they actually show fairly little overlap across CpG sites. There's really only a handful of CpG sites that overlap across these clocks. And, you know, in part, maybe that's to be expected. These clocks use different training phenotypes. They were, you know, trained using different tissues in terms of how the epigenetic profiling was done. So the original Horvath clock was a pan tissue clock. So they used 51 different tissues and cells. The majority have been done in whole blood. But these more recent clocks have also used plasma proteins, mortality, et cetera, time to death as their training phenotypes. And so these earlier clocks are starting to kind of become known in the literature as first generation clocks. These are clocks that use chronological age as their training phenotype. And these more recent clocks use phenotypic measures of age. And so, for example, the Levine clock here, she uses albumin, creatine, glucose, C-reactive protein levels, et cetera, 9 biomarkers in total as the training phenotype. And this most recent clock, the Dunedin Poem clock that was created by Dan Belsky, actually uses longitudinal epigenetic data from the Dunedin study to look at the rate of change across 18 different biomarkers. So it's actually looking at the pace of aging, a really neat clock. But so basically, what Morgan found using a number of different omics data was that these clocks are actually capturing really unique pathways of aging. And so I think this has a lot of implications for the work that I'm going to be showing you today with the social environment. So, while all this work was going on in geroscience, social scientists started to pick up on this and think, hey, well, this could be a really interesting way to get more at, mechanisms between the social environment and aging and health. I would say, so there's been a number of studies that have been done with SES and the clocks. And I think what's really interesting is that these associations across studies are just really not very consistent at all. The vast majority of studies have used first generation clocks, so the Horvath and Hnumm clock. And the Horvath clock in particular pretty much shows null associations in most studies, particularly with education. The Hnumm clock, which is profiled in blood, shows a little bit more promise there. Studies with second generation clocks have shown more consistent findings. And maybe this isn't surprising because they're trained on outcomes that may be more proximal to the social environment. So they might be picking up more on actual biological processes related to the social environment. But overall, I would say differences in study design, underlying sample characteristics, lack of statistical power, and positive publication bias really contribute to the kind of lack of coherent findings across studies. And I do want to mention a recent study by Eileen Crimmins and Jessica Fowl that just came out. And they are the only study to date I know of that has looked at associations between education and all 13 clocks. So most of them only look at 1 or 2 clocks. So in this study, what we did is we examined socioeconomic status associations across 2 large aging studies. And this is joint work with Jennifer Smith, Morgan Levine, Qiangxing Liu, and Wei Zhao. And specifically, we did a comparative analysis of associations between various dimensions of SES and 8 epigenetic clocks in 2 pretty large studies in terms of the sample size for epigenetics, MESA and HRS. HRS just recently released their epigenetic clocks online, so that's publicly available data that you can go download. And so we examined these different associations, and then we looked at the degree to which they're mediated by smoking, BMI, and alcohol consumption. These are health behaviors that really have a huge influence on the epigenome. And that, of course, we know also have quite a huge socioeconomic gradient, especially in high-income countries. In addition, what we did in this study, which was a little different, was we incorporated polygenic scores for epigenetic age acceleration to account for genetic heterogeneity and epigenetic age. So we started to look at how genomic contribution is factoring into these associations. And I also did not prepare what a polygenic score is for the same reasons that you cited, Patrick. So thanks for explaining that as well. And then, basically what we wanted to kind of get out of this study was just, see if across 2 large studies we can identify clocks that are more highly associated with SES, because we hope that this can contribute to future research on epigenetic pathways of aging and how to construct ones that are potentially more related to the social environment. So the data, as I mentioned, come from MESA and the HRS. MESA collected data, epigenetic data from monocytes on around 1,200 participants. MESA has a really nice distribution across ancestry groups, as you can see here. Mean age at blood collection was about 69. Health and Retirement Study, they collected epigenetic data from whole blood in 2016 in a random sample of the larger HRS. So this was a pilot study of about 4,000 participants. And they also have quite a nice distribution here in terms of ancestry groups and a similar mean age at blood collection as MESA. And notably, both samples are socioeconomically diverse. So 34% of MESA participants and 70% of HRS participants with DNA methylation data had at most a high school degree. These are some of the different SES exposure variables that we looked at. So education, highest degree obtained, household income in terms of quintiles, household wealth. Here we looked at whether an individual had assets in home or land, car, financial investments, occupational prestige, So here we merged in occupational prestige scores from the General Social Survey at the 3-digit occupational census level. And then we also constructed 2 different census tract level estimates of the neighborhood environment. One is measuring more socioeconomic disadvantage, so census estimates of education, occupation, income, wealth, poverty, employment, and housing. And then the social environment measures based on survey measures that are aggregated to the census tract that assess perception of the neighborhood environment. So aesthetic quality, safety, and social cohesion. And I'll show you, we did estimate each of these separately, but for the main analysis, we created an SES index that combines all of these aspects by taking the unweighted average across all of them. And basically for this index, higher values equal more disadvantage. I don't have time to go into all the nitty gritty about how we constructed the index and how we constructed all these different measures, but that's the gist of it. So here are the main results. This is the association between the epigenetic clocks and the SES index. So each of these is from a separate regression here of each clock. Here, the effect sizes are in terms of Pearson R estimates so that you can compare. They're standardized so that you can compare the estimates across clocks. And in model 1, which is the more saturated bar, that's this SES index coefficient is from a model that regressed the clock on the index age, sex, race, and various technical covariates. And then in model 2, which is the less saturated color, we added lifestyle factors, so obesity, drinking behavior, and smoking status. And what you'll notice is, and then here, just to mention the stars are Bonafroni corrected. So these pass Bonafroni correction in addition. And what you'll notice is that it's these 2nd generation clocks, again, that we see a really high association with SES. And also we do not see full mediation by health behaviors. So the socioeconomic gradient here in these clocks does not seem to be entirely explained by smoking. Then what we did was we looked at individual associations between each aspect of SES and the clocks. And we only did this in the 4 clocks that passed Bonafroni correction in the first step. And 2 things to notice here. I think, we do see some association with each measure of SES to a degree, some more so than others in the clocks. And the second thing that I found interesting was that, especially for education, income, and wealth, we see non-zero coefficient estimates for the most disadvantaged groups. So for those making less than $20,000 a year, for example, or those who have no assets and wealth, who have no degree, we see much faster epigenetic aging, here for those most disadvantaged groups compared to the others. And this is only in the HRS, I should mention. We then wanted to see, whether these results were driven by childhood socioeconomic status, since there's high correlations between the two, between own and childhood SES. And we found that wasn't the case. So here we were looking at maternal education. So, this first coefficient is the coefficient from model 2 of the SES index. The second coefficient adds mother's education to the model, and you can see that the estimate doesn't change much. And then this is the coefficient for mother's education. So higher mother's education, lower epigenetic age. What's interesting is that for the Grimm age and DNAm poem clocks, we really didn't find any association with mother's education. But overall, at least, we can see that there is an independent contribution of adult socioeconomic status. And finally, we wanted to look at the effect of a polygenic score. Unfortunately, there haven't been a whole lot of GWAS that have been done. There's one that's going to be released soon, but we weren't able to use it. That was a GWAS of some of these 2nd generation clocks. But for right now, we were kind of constrained to using GWAS on the 1st generation clocks. And in particular, this is a polygenic score that we used that was done for Horvath epigenetic age acceleration. And as you can imagine, the genetics that are contributing to Horvath aging are going to be different than the genetics that are contributing to the DNAm poem aging, because they are potentially capturing these different pathways of aging. But I think what we see here is just that, yes, the PGS is predictive. This is just, by the way, in the European ancestry subsample. We didn't find any evidence of moderation, which wasn't surprising. Perhaps, I think we might be kind of underpowered, especially because we don't see an SES index association with the Horvath clock. But I think what this is showing is that, there is independent association genetically. The genetics are also contributing to faster epigenetic aging. In addition, like especially here, we can see with the SES index. So in terms of takeaways from this study, we found that second generation clocks trained on phenotypic markers of aging are more correlated with the social environment, perhaps because the aging phenotypes that they were trained on are more proximal to the social environment. We found that education and income in particular display the most robust associations with the clocks, particularly for individuals in the most disadvantaged groups. And I didn't mention this on that slide, but we did also then put all the SES factors into the same model to see what survived, because of course they're all highly correlated, and it was mostly education and income. Associations were partially but not fully mediated by lifestyle. So we saw that differences in health behaviors alone cannot explain the SES gradient in epigenetic aging. And finally, that genetic risk, in addition to social disadvantage, may contribute additively to faster epigenetic aging. In terms of what's next in social epigenomics, I think, from the work that I've done here, what I've really seen is that the study populations and the phenotypes that are used to train these clocks are really key to illuminating social pathways of aging. So, as epigenetic samples get larger, we can actually construct epigenetic biomarkers that capture unique pathways of aging in specific environmental contexts. So we could actually construct, epigenetic biomarkers that are trained on social outcomes, or on other phenotypes that are really highly associated with the social environment. And then we can deconstruct those clocks, right? That we can pull them apart and look at the CPG sites that were selected and the potential genetic pathways that they're capturing. So in this way, we can kind of back out the social environment. One example from my ongoing work is I'm working with Hans-Peter Kohler and Ileana Kohler at collecting genetic and epigenetic data in the Malawi longitudinal study of families and health. This is a distinctly low-income context where individuals are living on less than $1 a day. And so my hypothesis here is that the clocks that were constructed in a really high-income context are just not going to replicate in this low-income context, that we're going to find really unique environmental, clock associations. I also think there's a huge need for causal work in social epigenomics. And I think this is in part because we just haven't had large enough samples and not population representative samples where we can really exploit natural experiments. This is changing. And for example, with the HRS, I have ongoing work with Patrick Turley and Dan Benjamin and David Cesarini, where we're looking at the causal impacts of smoking behavior on epigenetic signatures using state year level variation and cigarette taxation. So that's just one example of how we can maybe move the work forward in terms of population level work. And finally, one thing that I'm really excited about is using epigenetic annotation to construct tissue-specific polygenic scores. I think this is a really exciting tool for looking at mechanisms behind gene-environment interactions. And I have some ongoing work with Kyung-Shi Liu, where we're looking at the impact of smoking and alcohol abuse on ADRD, using tissue-specific polygenic scores for the liver and for monocytes. So I want to say a huge thank you to my collaborators and my students, in particular for the studies that I mentioned today, just wouldn't be possible without them. So this is definitely team science, definitely a team effort. So thanks, and please feel free to reach out with questions, comments via email, and yeah, thanks for listening.